Today's lecture is the first in a two-part series dealing specifically with damage of DNA and the repair of that damage and how it occurs at the cellular level. The first thing that I want to start with or address is the definition of a mutation. A mutation typically has a negative connotation associated with it, but at the basic level in molecular genetics, it's defined as a process that produces an alteration in DNA sequence or chromosome structure. This can be lots of different types of modifications, like differences in the number of chromosomes, deletion of chromosomes, duplication of chromosomes, or rearrangement in chromosome structure. However, when we're talking about molecular genetics, we oftentimes focus on the changes that occur at the DNA sequence level, in addition to those associated with chromosome structure. But before we start talking about specific types of damage and how they are repaired, I want to actually classify mutations for you, or discuss the fact that mutations can be classified in lots of different ways. One way to classify mutation is whether or not it's considered spontaneous versus being induced. A spontaneous mutation is one that occurs, quote-unquote, naturally. It's a naturally occurring mutation, and it's not induced by any specific mutagenic or chemical agent. It's just something that happens, for lack of a better way of saying it. That's in contrast to something referred to as an induced mutation. And an induced mutation is one that results from the influence of an artificial factor of some sort. Another way that mutations can be classified is whether they are gametic mutations versus somatic mutations. Now, a somatic mutation is one that occurs in a somatic cell, and this is usually masked in the individual. Somatic cells are cells that are different or distinct from gametes, and gametes are the sex cells that allow for the transmission of genetic information to the next generation. So if a mutation occurs in a somatic cell, that is not going to be typically passed on to the next generation. It will only have the effects in the individual that the mutation occurs in. Something a lot more serious might be something that's referred to as a gametic mutation. A gametic mutation occurs in the genes or chromosomes found within the gametes or gamete-forming tissue. This then has the potential of being passed on from one generation to the next. So if you look at the slide with point number 1, we have a somatic mutation occurring in a non-reproductive type of cell in this particular fish. That somatic mutation can be amplified through mitosis as the organism continues to grow, and you have more cells that have that particular mutation. So in this case, you have a population of mutant cells, but in the long run will only potentially cause problem to that individual and will not be passed on to the next generation. Along the bottom of the slide, you see the alternative example where you may have a mutation that occurs in germline tissue, in gametes or gamete-forming tissues. In this particular case, that mutation would be passed on to the next generation, and those mutations then are carried on to other individuals that result from this particular individual. And so this might be something a lot more serious because of the longevity and passing on from one generation to the next. So the first two slides dealt specifically with categorization. These types of mutations were exactly what we discussed at the tail end of the last lecture. Base substitutions, like you see in this table, result in a change for one nucleotide for another. We discussed also at the tail end of last lecture that those can have different effects. A substitution can result in what's known as a silent mutation, and that silent mutation results in no change in amino acid, because of the redundancy of the genetic code. Substitutions can also fall into the class or category of being a missense mutation. And in a missense mutation, This substitution results in a different amino acid being inserted than what was originally coded for. So the single nucleotide change results in a different amino acid being inserted during the protein synthesis process. And lastly, a substitution can be categorized as a nonsense mutation. And a nonsense mutation is where the codon has been altered to become a stop codon. This is considered nonsense because what happens during protein synthesis is a premature termination of the protein synthesis process because the original codon has now been changed to a stop codon and protein synthesis stops. Now, insertions or deletions can actually be much more detrimental when we are talking about the downstream effects as far as protein synthesis is concerned. The insertion of a nucleotide or the deletion of one or more nucleotides will then alter the reading frame in the protein synthesis process. If you recall, protein synthesis again starts at the AUG codon, and this sets up what's known as the reading frame. From that point on, every 3 nucleotides is a codon and codes for the insertion of a specific amino acid. If you have now inserted an additional nucleotide or deleted a nucleotide, the entire reading frame has been shifted, and what results is a protein that is essentially gobbledygook, doesn't make sense, and oftentimes results in premature termination. A third way that point mutations can occur is what's referred to as expanding nucleotide repeats. There are certain genes that have codons that are repeated multiple times within the gene in their normal circumstance. So if you look at this table, we have a number of different disorders that are listed here where the number of repeats of a given codon is altered from the normal range. So let's look at the first example, which is spinal and bulbar muscular atrophy. So the repeat sequence is CAG, and in the normal copy or the normal allele, there are somewhere between 11 to 33 repeats of CAG. In individuals with this particular type of muscular atrophy, there is a dramatic increase in the number of these particular repeats, anywhere from 40 to 62. And you can look through this table and see multiple examples of that overall. This typically occurs with problems due to replication errors. The best analogy that I can give for how this happens is that if you think about reading a sentence, us as individuals reading a sentence, and if the sentence had the word the, a 3-letter word, repeated over and over and over again, as you're reading that sentence with the word the, you might lose your spot and accidentally start all over again. And this is exactly what happens during the replication process, is the enzymes sort of lose their place in the process and begin to add in additional repeats than what are normally called for. Okay, so now let's transition a little bit into what actually could be causing these mutations that we just discussed in the previous few slides. One example of how a mutation can in fact occur is what's known as a Tautomeric shift. A Tautomeric shift is a mutation that results from a shift in the localization of a hydrogen atom. Essentially what that means is that isomers can form with specific nucleotides. So there can be isomer formation with specific nucleotides. And when these isomers do in fact form, it allows for an incorrect type of base pairing to take place. So let's look at the examples that we see in this slide or this figure. In panel A, you're looking at the standard base pairing arrangement that we know exists. The base pairing rule, the sequence complementarity that we've talked about multiple times throughout the several past lectures. Thymine, always hydrogen bonding with adenine, and specifically they fit together like puzzle pieces with 2 hydrogen bonds allowing for that interaction. On the right-hand side in panel A, you see cytosine, hydrogen bonding with guanine. Again, this is our normal, typical base pairing that takes place with 3 hydrogen bonds that fit together like puzzle pieces to allow cytosine and guanine to interact. However, in panel B, what you can see is these tautomeric shifts. They are a shift in the localization of a hydrogen atom, resulting in an isomer to form. So what you see with thymine is that this isomer now allows for 3 hydrogen bonds to form instead of 2. And specifically, that allows for base pairing with guanine. You can see how this could be a potential problem during DNA replication. If the template sequence has a T, then the newly synthesized DNA should get an A as the complementary sequence. However, if that T has undergone a tautomeric shift, now the replication machinery allows for the base pairing of T and G, and now we've inserted an incorrect nucleotide during the replication process. And this is how a base substitution can occur, specifically in this category of a tautomeric shift. And a similar example can be seen on the right with cytosine undergoing the tautomeric shift, allowing now for 2 hydrogen bonds to form, which then allows it to base pair with adenine rather than guanine. Another cause of mutations, specifically insertions and deletions, is due to what's referred to as mispairing. This too is a problem that occurs during DNA replication, specifically when there is a stretch of nucleotides that are exactly the same. So in this example, at the very top of the slide, you see the template strand, and it has a stretch of T's, one right after the other, right smack dab in the middle of that sequence. On the top, you see the newly synthesized strand, which would then have a stretch of A's because of the sequence complementarity. However, there could be an error with that newly synthesized sequence where a slight loop occurs where one of the A's doesn't base pair anymore or is mismatched. And you can see that little blip in portion or panel 1 of this figure. If that does in fact occur, if you follow that through the replication process, there is this blip that remains in the sequence overall. This would then result in the addition of a nucleotide in the newly synthesized strand. Alternatively, if you look at panels 3 and 4, you could have the looping out occur on the template strand rather than on the newly synthesized strand. And again, these are all mistakes or errors that could in fact happen. Don't necessarily always happen, but could happen simply by those hydrogen bonds releasing and falling back in the incorrect position. If you follow that through to panel 4, you would see that there is a omission of a nucleotide, a deletion of a nucleotide that has occurred in the newly synthesized strand. So in either one of these situations, there is a problem that has occurred, either the insertion of an additional nucleotide or the deletion of a nucleotide. Another cause of mutations would be the insertion of a base analog. A base analog is a molecule that can be substituted for either a purine or a pyrimidine during the nucleic acid synthesis process. What happens if this particular analog gets inserted is you now have a nucleotide that has a base that looks very similar to one of the quote-unquote normal bases and could potentially be inserted during the replication process. The example that you see here is a analog known as 5-bromouracil. 5-bromouracil gets mistaken for the base thymine and can in fact get incorporated into a nucleotide. If it is incorporated into a nucleotide, when replication occurs, what you can see on the right-hand side of this figure is that What would base pair now with 5-bromouracil is guanine. So again, 5-bromouracil is an analog for thymine. It structurally is very similar to thymine, so it would be inserted as a thymine would. However, when replication takes place, you can see that you have the incorrect type of base pairing that would now occur. And this is just one specific example of a base analog. There are other examples of molecules that, again, are structurally very similar to other bases and can get mistakenly incorporated into a nucleotide. And if that happens, that nucleotide with an incorrect base could be incorporated into the DNA, hence the mispairing that might occur during DNA replication. Another way that mutations can in fact occur is at the time of crossing over or genetic recombination. If homologous chromosomes misalign, that means they don't align the way they're supposed to during the crossover or genetic recombination process, you can have something that's referred to as unequal crossing over. And with unequal crossing over, you have resulting chromosome structure that has different genetic information in it than it did previously. And so here too, you can have insertion or deletion of stretches of nucleotides with mistakes occurring during the crossover or genetic recombination process. Other ways that point mutations can in fact take place. Alkylation is yet another way that mutations can in fact be induced. Alkylation is defined as the addition of short hydrocarbons being transferred to reactive sites on bases. These short hydrocarbons are typically a methyl group or ethyl group, with 1 carbon for methyl groups and 2 carbons for ethyl groups. And there are chemicals that typically cause this alkylation to take place. The end result of alkylation is mis-base pairing. So in the top figure, you see the correct or normal type of base pairing we would see between adenine and thymine and guanine and cytosine. In the bottom figure, you can see that guanine has been alkylated by a chemical agent known as EMS, which is short for ethyl methane sulfonate, also known as mustard gas. And in this case, it does in fact change the structure, the molecular structure of guanine to become 6-ethyl guanine. When this happens, this causes mis-base pairing to take place. Again, what it should be is a G. That G is changed structurally to become ethyl guanine, which now only base pairs with thymine. The next example is a category of causes that are due to chemical mutations. Chemical mutagens. Chemical mutagens are chemicals that can cause DNA damage. They change the DNA in some way, and that can result in detrimental downstream effects overall. What you see here is an example of several different mutagens and what exactly is happening when that mutagen is damaging or altering the structure of the DNA. What you see in this example in panel A is guanine as the original base. If it is treated with a specific mutagen known as EMS, you end up with alkylation, which changes the structure of guanine. With that structural change, again, you have a mispairing event that will take place. Guanine normally hydrogen bonds with cytosine. However, if it has been mutated with this alkylating agent, it will now hydrogen bond with thymine. And you can see the other examples in this particular slide as well. So in the last slide, you saw chemical mutagens that can cause changes in base structure, which eventually results in mispairing events that can take place. There are other chemical mutagens that physically cause damage to the DNA. These are things that result in destruction overall of the structural integrity of the DNA molecule. What you see on the slide are a number of molecules that actually can bind and get in between the bases in the DNA structure, like you see in the right-hand panel. And when it does that, it actually causes a distortion of the helical shape. This makes a very unstable type of DNA molecule, and also a molecule that is very hard to replicate. The last two examples of causes of DNA mutations are related to one another in that they both cause structural damage of the DNA. The first example, labeled number 7, is UV light. Ultraviolet light does in fact have a great deal of energy associated with it. In particular, if there is a DNA sequence that has two thymines, one right next to each other, the exposure to UV light can cause what's known as a thymine-thymine dimer. And if you look at this figure, you can see that what happens is structurally there is covalent bonds that have now formed between these neighboring thymines. This then causes a structural distortion in the DNA. And then lastly, what you see as number 8 is ionizing radiation. These would be things like X-rays or gamma rays. These typically destroy the connection between the nucleotides, like you see labeled here with the phosphodiester bond. And with the breakage of these bonds, this then causes, again, physical damage in the DNA.